just give you an example. So in C. elegans, most genes were originally predicted using gene-finding algorithms. So algorithms that would go in and look at a sequence, at a chromosomal sequence, and pick out all of the coding sequences. This was one of the gene models. In the last year and a half, it has changed from its original model. The original model had the stop codon in this exon. The exon was a little bit bigger, and there was a stop codon, and there was no exon out here. So this is a pretty big gene. And one of the interesting things, if you go into this gene, and we can just click on this location, and it's going to expand and go to a genome browser. Now, if you go to Flybase, there are genome browsers that look almost exactly like what you see in Wormbase or any of the databases for the model organisms. I think I mentioned that because C. elegans was the first model organism to have its genome sequenced, all of the others kind of copy the database development that's gone on in C. elegans. So what we see here, I've already opted for all of these different, what we call tracks. So here's a gene model, and you can see the gene model predicts the exons. There's all kinds of other things that you can look at, transposon insertions that people have done. There's a group making what they're really ambitious. They're going to make 1 million mutations. Actually, they're going to easily make 1 million mutations. It's a very simple process they're doing. So it's not as ambitious as they would like you to believe. But down here, ESTs aligned by essentially a BLAST-like program. ESTs stand for express sequence tags. It's bigger. Step in. EST for expressed sequence tag, which is the same thing as saying RNA, or what you might refer to as cDNA, which is essentially RNA that has been reverse transcribed to form complementary DNA. How many people are familiar with those terms? Okay, not everybody. So RNA can be reverse transcribed using reverse transcriptase to make cDNA. So cDNA is simply the complement of RNA molecules. And when people go out and in every model organism, there have been labs here and there that just go in and start sequencing RNA molecules kind of at random. And then they upload those sequences so that other people can study them. So here is, for instance, a sequence of RNA that was uploaded. It's given a little descriptor. And so you can see here that this cDNA, or EST, or really RNA sequence, tends to support the gene model that's found here. So we can see that this exon has clearly been found in RNA from a cell. Supports this gene model as being correct. Or originally, this gene model ended about right here, didn't have this end portion, and so this gene model is now correct based on the EST sequence here. So in fact, if you were to come up with DNA sequence, a sequence that's probably being expressed, you can go into these databases and you can pretty quickly say what's actually probably the expressed sequence in the organism itself by finding ESTs or cDNAs that are there. So it's a pretty rapid way of doing this. And again, most of the model organisms have this. All right, so let's go back to these sequences and talk more about what we can do with these sequences once we have an amino acid sequence. So the last time, what did we do to analyze them? Blast, right? We blasted them. We blasted them. And we found sequences. We found a whole bunch of sequences. And what did we learn from that blast? What did that give us?
Showed it was in a bunch of different organisms.
What's that?
Showed it was in a bunch of different organisms. That's
Okay, so we learned similar sequences in other organisms. Typically, we call those homologs. Right? We've used the term homolog before to refer to a homologous chromosome, right? Every individual that's diploid has 2 copies of each chromosome. We call those homologs. Here, a homolog means a gene that has a very similar sequence in another organism. Homolog typically means, if we say homolog, what we mean is that at some point in evolutionary history, we have 2 forms of the same gene. Say a C. elegans form of some gene, let's say RFT1, and some form human RFVT3. I don't know if you can see that over on the edge of the class, but that at some point in evolutionary history, there was an organism that had 1 copy of that gene, and that copy has split, and now C. elegans has it, and humans have it. And these are actually 2 genes, the human riboflavin vitamin transporter 3, the C. elegans RFT1, the riboflavin transporter. That's the one I showed you. This gene right here is the C. elegans version of the human riboflavin transporter. So these are what we would call homologs. We think they're homologs. We don't know for sure. We can't go back in evolutionary history and find this organism that has it. So this is just a hypothesis just because they're similar, but we call them homologs. You might also see that there are what we call paralogs out there. So a homolog are sequences that we find that are similar in different species. Paralogs, does anybody know what a paralog is?
Similar in the same species.
Similar in the same species. That's right. So a paralog is one that's similar in the same species. So if I come back to this page, and I can go down here, and this page gives me, without even having to do the blast search, and again, most of the model organisms, their database, their web databases have this. You could go across here, and you can see all of the homologs. I said HRFVT3. That actually, this is old. That has actually been updated now. So you see the best homologs. Humans, there's a bunch in humans. And if we look down at the bottom of the page, it says paralogs. And we see that C. elegans has another gene that's very similar to it. In fact, C. elegans has 2 riboflavin transporters. Humans have 3. So humans have 3 paralogs. C. elegans has only 2. I didn't mention it, but you see ortholog. An ortholog. So ortholog really specifically means the ones that are in different species. Paralogs are the ones that are in the same species, but typically we refer to them as homologs. So there we go. We've got all of these genes out there. So we can find similar genes. But there's more that we can do. Remember when we were doing that BLAST search, we also came up with some ideas about function. Right? Because we can see from the genes that we pull up, they're given names in most cases based on their function. Right? Do you remember the function that we saw in the gene names the last time? Only 2 days ago. Presenilin was the name, and that's involved, that gene is involved in early onset Alzheimer's, so its protein has a function in brains, and when you knock it out, or when it's affected by mutation, you get senility, early onset senility. So that's this gene right here. In humans, the homolog is for senility. Now, one of the things, when you get all of those sequences, another thing that you can do after blasting them is you can take them and you can do an alignment. You can align the sequences. There are programs that will allow you to take multiple sequences and try to figure out the positions at which they share similarity and align them. Here's an alignment, a portion of that SPE4 gene. And so we look down here, and there's an alignment with close species, close relatives to C. elegans, C. ramanii, C. brenneri, C. briggsii. Then farther afield, there's Ascaris suum, Christianicus pacificus. So we're looking here at all of the nematodes, and then here's C. elegans cell 12, so this is a paralog. And so what we're doing here is every place where you can see some shading, we've got similar amino acids. So in this position, there's all glycines in various places along here. What you get a feeling for is when you have a position, an amino acid position, that's highly conserved, okay? Meaning that position, well, I've erased my tree, but that position probably had the same amino acid at that position way back in evolutionary time before the split of those genes. So when we have a position that's highly conserved, that position is very, very important for function. So conserved positions are important for function. And if you think about this, it makes sense, right? You've got a position. These positions, we know DNA is being mutated at a pretty rapid clip out there in nature over evolutionary time. Pretty much every amino acid position is going to be changed by mutation in individuals. And because these have remained exactly the same in all of these different species, that means every individual that was a mutant that had an amino acid position change here